Hello, in this video we will discuss about the glucagon. So the glucagon is the endocrine hormone which that is released from the pancreas. So we will discuss about this. So here is the during the fasting time the glucagon will be activated. So it means when we will not eat the carbohydrate and protein and fat, basically all food. So the during the fasted state the glucagon will be produced. This glucagon is basically is the a destructive hormone, not instructive hormone, or it is the catabolic hormone, which that is used for the catabolism of the body, as compared to the insulin, which that is the anabolic hormone. So here is the pancreas, which that is present with the duodenum, and here is the pancreas, which that is contain a pancreatic acini. It is also known as islet of Langerhans, that contain a beta cell, delta cell, and alpha cell. Beta cell is used for the produce insulin, while the alpha cell is produced for the glucagon, and the delta cell, which that is produce the somatostatin, which that is the regulate the beta cell and alpha cell. So here we will look at the alpha cell, which that is during the low glycemic index in the or low glycemic uh, time during the fasting state. This glucose will be enter, and the minor glucose will decrease the ATP. Why? Because the less glucose will decrease the ATP due to the glycolysis will be less and Krebs cycle will be less, and which that is stimulated by the mechanism we will discuss later in this video to stimulate the glucagon, and that glucagon will be moved into the blood. So that is why it is known as the endocrine, not exocrine. So here is the hypoglycemia when during the fasting state. So here I am drawing the muscle and liver and the adipose as a adipose cell and muscle cell, as well as the muscle cell. So basically adipose tissue contain a adipocyte, while the livers contain a hepatocyte, and the myocyte, which that is present in the muscle. So when glucagon will be attached with the glucagon receptor, so after this receptor the lipolysis, ketogenesis, and gluconeogenesis and glucogenolysis, which that is the glycogen will be breakdown. So the breakdown of free fatty acid will be converted from the triacylglycerol to free fatty acid, and the adipose tissue also contain glycogen but minor quantity. So here is the gluconeogenesis and glycogenolysis, and gluconeogenesis and proteolysis will be occur in the hepatocyte. While on the other hand, the muscle cell will not a receptor of the glucagon, but the proteolysis is occur due to the fasted state, but not due to the glucagon. So here is a glucose will be increase in the liver, and this glucose is basically the glycogenolysis will increase the glucose, and that means the catabolism of the glucose by acetyl CoA and ATP production, as well as the lipolysis to result of the free fatty acid. And the acetyl CoA also convert the new sugar type is known as the ketones. This ketone is known contain keto acids from amino acid, and acetoacetate and beta hydroxybutyrate, and acetone and acetaldehyde. Basically the beta hydroxybutyrate is the important major. While on the other hand, the proteolysis will produce the protein, apoprotein A, E, and D. While on the other hand, the free fatty acid, which that is also accumulate and cholesterol to produce a very low density lipoprotein, which that is transported into the blood circulation for transport the fatty acid into the muscle. So that was the overview. So let's begin to understand with the whole detail of the alpha cell. So here I am drawing the pancreas, which contain a alpha cell. This alpha cell we will magnify this alpha cell or zoom this alpha cell to watch the whole detail and mechanism of the production of glucagon basically. So you should concentrate on the production of the glucagon just. So remember about that. We are studying about the releasing of the glucagon during the fast state or during the catabolic state of the metabolism. So here is the receptor. This receptor is known as the G-couple receptor as well as the adenylate cyclase enzyme, which that is a protein and are present in the plasma membrane. While the G-couple receptor is G protein couple receptor, which that is attached with the subunit of the protein alpha, beta and gamma. So here is basically I am drawing the digestive system which contain a duodenum. This duodenum is contain a lumen and enteroendocrine cells, which that is produce the hormone endocrinally into the blood circulation, and this can be a GIP. So this GIP will be bind with the GIP receptor, which that is a G couple receptor. After the binding of the GIP, we will discuss what that means and what is the happening in the intracellular signaling. So here is the G couple receptor. We receive the GIP as well as basically another hormone we will not discuss in here. So that's alpha subunit contain a GDP, guanosine diphosphate, which that is activated by the phosphokinase enzyme to produce a GTP alpha, guanosine triphosphate, and this guanosine triphosphate will be active and will go into the adenylate cyclase. Cyclase means the cyclic AMP will be produced from the adenylate cyclase enzyme. So that's ATP used to produce cyclic AMP. After the production of cyclic AMP, basically the pyrophosphate will be released and the cyclic AMP will be activate the inactive pKa, phosphokinase A, protein kinase A, and this protein kinase A will be active by the phosphorylation, and that's cascade of the phosphorylation to become a transcriptional factor. This transcription factor in the cytoplasm in the form of protein, and this protein will be phosphorylated to a specific signal. This specific signal will go into the nucleus by a nuclear pore, and that's go by the addition of the primer and the addition of the phosphatase enzyme, phosphate group, and on the other hand the addition of the helicase enzyme where the gene cluster, for example the glucagon gene cluster, and that transcription will begin from the primer to and polymerase enzyme. This polymerase enzyme basically is the produce a messenger RNA. This messenger RNA is the pre-messenger RNA. After the splicing will be intron will be released and exon will be left behind, and that's messenger RNA will contain a poly A tail and cap. This polysome formation, when the ribosome will be bind collectively, that is known as the polysome. This polysome will be produce the polypeptide from the transfer RNA, and this transfer RNA will be produce a protein is the polypeptide. So the initiation factor is the basically bind and initiate the methionine first is the AUG codon with the anticodon of the transfer RNA, and that produce the polypeptide, and these polypeptides will be terminate and produce a pre-pro-glucagon. These pre-pro-glucagon contain a N and N carboxyl terminus and N terminus. So this is the pro-glucagon will be produced after the splicing, and that's enzyme used for it, and that's convert after the restriction to GRPP and pro-glucagon fragments. On the other hand, the major fragments is known as the 29 amino acids containing polypeptide. This is the major peptides which that is used for the go into the Golgi, and that's go into the Golgi to the glucagon containing mature vesicle for exocytosis will be formed. This mature vesicle containing glucagon is required the mechanism for exocytosis into the bloodstream. So in this time we are understanding the mechanism for the releasing of the vesicle into the bloodstream. This exocytosis we will discuss first. So here is the hypoglycemia will be occur in the bloodstream. So the low level of the glucose will be moved by the GLUT4 channel, and thus glucose will be entered into the cytoplasm from the bloodstream, and the glycolysis will be occur and produce ATP. After all, the pyruvate will be formed. This pyruvate will be moved into the mitochondria to produce acetyl-CoA. 2-acetyl-CoA will be produced from the 1-glucose due to the 2-pyruvate production. And the citric acid cycle run the citric acid cycle and electron transport chain to produce ATP. The ratio of ADP will be decreased as compared to the high hyperglycemia. So the hyperglycemia will lead to produce the insulin, while the hypoglycemia will produce the in the mitochondria, which that is produce the stimulate the K ATP potassium ATP channel, which that is efflux of the potassium outside of the cell to create a positive charge inside, while the outside is a negative charge. So here is the T-type channel, calcium channel, which that is the fast and basically is the limited calcium will be moving to create a more positive charge as compared to the outside. It means the changes of the positive charge inside. So the next one is basically is the sodium will be moved in also from the sodium channel, and the calcium L type and N type channel is the basically long lasting normal channel, which that's create more positive charge inside. This more positive charge inside will create the triggering of the glucagon containing mature vesicle for exocytosis, and that's exocytosis will begin. So this exocytosis of the glucagon will be released the glucagon into the blood vessel, into the blood circulatory system, and ultimately will reach into the their target, for example liver and adipose tissue. When bind with the adipose tissue, that's glucagon will increase the glyconeogenesis, glycogenolysis, and glucose increase, and acetyl-CoA production and free fatty acid, as well as the adipose tissue also look like this function. So thanks for watching. Please make sure to subscribe, like, and share this video to your friends.